Have you ever wondered how TALEN works? Let's dive in. TALEN, short for transcription activator-like effector nucleus, is a revolutionary tool in the world of genome editing. This fascinating technology stems from the plant pathogen Xanthomonas. The DNA binding domain of these TAL effectors, derived from Xanthomonas, consists of a variable number of amino acid repeats, with each repeat recognizing a single DNA base pair. This recognition is made possible through two hypervariable amino acid residues known as repeat variable diresidues, or RVDs, located at positions 12 and 13 within each repeat. These TAL effector repeats can be assembled modularly, allowing the creation of a TAL protein that recognizes a specific target DNA sequence. The process is quite straightforward and can be customized to target any locus in the genome. So, TALEN is a powerful tool that allows scientists to edit genomes with high precision. So how does TALEN recognize specific DNA sequences? Well, it all comes down to the DNA binding domain of TALs, derived from Xanthomonas TAL effectors. This domain is comprised of a variable number of amino acid repeats, with each repeat containing 33 to 35 amino acids and recognizing a single DNA base pair. The DNA recognition happens through two hypervariable amino acid residues located at positions 12 and 13 within each repeat. These are known as repeat variable diresidues, or RVDs for short. Now here's where it gets interesting. TAL effector repeats can be assembled in a modular fashion. By varying the RVDs, we can create a TAL protein that recognizes a specific target DNA sequence. The process of linking these repeats is quite straightforward. And with this method, long TAL effectors can be designed to target any locus in the genome. This means TALEN can be programmed to bind to virtually any DNA sequence. But what happens once TALEN binds to a target DNA sequence? Well, this is where the fork-1 nucleus comes into play. Acting like a pair of molecular scissors, the fork-1 nucleus, fused to the TAL proteins, creates double-stranded breaks in the DNA at the specified genomic locus. Now, you might be wondering, why use a pair of TAL proteins? The answer lies in reducing off-target effects. By using two TAL proteins, each recognizing a different sequence, we ensure that the fork-1 nucleus only cuts when both TAL proteins are bound to their respective DNA sequences. This dual recognition system significantly minimizes the chances of the nucleus making a cut at an unintended location, thus enhancing the specificity and accuracy of TALEN. So, in essence, the fork-1 nucleus, coupled with the TAL proteins, allows us to precisely target any locus in the genome and make a clean, double-stranded break. This is how TALEN ensures accuracy in genome editing. What happens after these DNA breaks are created? Well, the cell jumps into action with two primary repair mechanisms: non-homologous end joining, or NHEJ, and homology-directed repair, also known as HDR. Let's start with NHEJ. This process is a bit of a wild card. It's quick, yes, but it's also prone to errors. Imagine it like a hurried handyman fixing a broken pipe with whatever's at hand, not caring too much about precision. When NHEJ happens within the coding sequence of a protein coding gene, it often introduces what we call a frameshift mutation, effectively silencing the gene. On the other hand, HDR is the careful, meticulous carpenter. It uses what we call donor sequences, essentially blueprints of the correct DNA sequence, to introduce a defined new DNA sequence. This makes HDR a powerful tool for precise genome editing. Thus, TALEN not only breaks DNA, but also guides its repair. So let's quickly summarize how TALEN works. The DNA binding domain of TALs, derived from Xanthomonas TAL effectors, is composed of a variable number of amino acid repeats. Each repeat, recognizing a single DNA base pair, contains 33 to 35 amino acids. The DNA recognition process is facilitated by repeat variable diresidues, or RVDs, located at positions 12 and 13 within each repeat. These TAL effector repeats can be assembled in a modular fashion, adjusting the RVDs to create a TAL protein that recognizes a specific target DNA sequence. With the help of a pair of gene-art TAL proteins fused to the FOC1 nucleus, double-stranded DNA breaks can be created at your chosen genomic locus, minimizing off-target effects. The breaks induced by the FOC1 nucleus domain are then repaired through either non-homologous end joining, prone to errors, and often resulting in a frame shift mutation, or homology-directed repair, which can introduce a new DNA sequence. And that's how TALEN, a revolutionary tool in genome editing, operates at a molecular level.